अस्सलाम वालेकुम स्टूडेंट्स, होप यू ऑल आर फाइन। स्टूडेंट्स, आज हम बायो क्लास 11 चैप्टर नंबर 11 एक्सरसाइज लॉन्ग क्वेश्चन आंसर सॉल्व करेंगे। सो लेट्स स्टार्ट। फर्स्ट क्वेश्चन हमारे पास, डिस्क्राइब स्ट्रक्चर ऑफ ह्यूमन मेल रिप्रोडक्टिव सिस्टम, आइडेंटिफाई फंक्शन ऑफ ईच पार्ट। The human male reproductive system consists of following structures. First one is sternal genitalia, and internal genitalia. It includes pair of glands function, testosterone production. Next is epididymis, tube-like structure, function, storage. Vas deferens, ductus deferens. Muscular tube transporting the urethra, function, transport. Seminal vesicles, glands producing seminal fluid, function, seminal fluid production. Prostate gland, gland producing prostatic fluid, function, prostatic fluid production. Next are accessory structures. Sertoli cells, spore cells in the seminiferous tubules. Function, nourishment and support. Next are Leydig cells, cells producing testosterone. Next are Cowper's gland, bulbourethral glands, gland producing trajectory fluid. Next are Next question is, explain the principal reproductive hormones of human male and explain their role. The principal reproductive hormones in humans are testosterone, produced by Leydig cells. Role: regulates spermatogenesis, develop and maintain reproductive organs, stimulates libido. Next is follicle stimulating hormone FSH, produced by pituitary gland. Role: stimulates spermatogenesis, regulates Sertoli cell function, nourishes. Next is luteinizing hormone LH, produced by pituitary gland. Role: stimulates Leydig cell. Next is inhibin, produced by Sertoli cells. Role: regulate FSH production, negative feedback, and inhibits spermatogenesis. Next is prolactin, produced by pituitary gland. Role: inhibit GnRH, gonadotrophin releasing hormone production. These hormones work together to regulate reproductive system. Hormonal balance is crucial for reproductive health. Imbalances can lead to issues like dysfunction or infertility. Next is question number 3. Explain structures of human female reproductive system. Identify function of each part. The human female reproductive system consists of following structures. Ovaries. Small oval-shaped organs located in pelvis responsible for producing eggs, oocytes, and releasing hormones, estrogen and progesterone. Next is fallopian tubes. Two narrow tubes connecting the ovary to the uterus is possible for transporting eggs from ovaries to the uterus. Fertilization typically occurs here. Next is uterus. It is possible for supporting fetal development and contracting. Next is cervix. Lower part of the uterus possible for producing mucus and. Next is vulva. It includes labia majora and labia minora, clitoris. Each part work together to support reproductive functions such as menstruation, etc. Next question is describe your menstrual cycle and explain its hormonal regulation. The menstrual cycle is a complex process controlled by hormones. Is a step-by-step explanation of the events and hormonal regulation. First of all, events of menstrual cycle. Menstruation, day 1 to 5. Luteal lining shades, result follicular phase. Next is follicular phase, day 6 to 14. Already use follicles and estrogen level rise. Next is ovulation, day 14. Pituitary gland releases LH, luteinizing hormone, causing ovulation. Next is luteal phase, days 15 to 28. Corpus luteum forms, producing progesterone. Next is premenstrual phase, day 6 to 28. Progesterone level drop, preparing for menstruation. Hormonal regulation. First one is gonadotrophic releasing hormone GnRH, released by hypothalamus, stimulate pituitary gland. Next is follicle stimulating hormone FSH, released by pituitary gland, stimulate follicle growth and estrogen production. Next is luteinizing hormone LH, released by pituitary gland, triggering evolution and corpus luteum formation. Estrogen, produced by follicles, stimulate uterine lining growth and preparing for ovulation. Next is progesterone, produced by corpus luteum, maintaining uterine lining and preparing for implantation. Feedback mechanisms. Estrogen and progesterone level feedback to the hypothalamus and pituitary gland, regulating gonadotrophic releasing hormone, follicle stimulating hormone, and luteinizing hormone release. Low estrogen levels stimulate GnRH and FS release, promoting follicle growth. High estrogen level inhibit GnRH and FSH release, preventing excessive follicle growth. Progesterone level regulate LH release, ensuring timely ovulation. This intricate hormonal balance ensure a regular menstrual cycle. Next question is, what is infertility? What is the causes of male and female infertility? It is the instability. It is caused by various factors. First one is low count oligospermia or poor motility astenospermia. Next is abnormal morphology, tritospermia, hormonal imbalances. For example, low levels of testosterone, very causal large wages scrotum infections, for example, epididymitis or chitis, blockage of ducts, for example, vas deferens, genetic disorders like Klinefelter syndrome, lifestyle factors e.g. smoking, excessive alcohol, stress, obesity, etc. Next are ovulation disorders, for example, polycystic ovary syndrome or PCOS, tubal damage or blockage due to surgery, infection, or endometriosis. Uterine or cervical abnormalities like fibroids or polyps, hormonal imbalances like thyroid disorders, hyperprolactinemia, endometriosis, adenomyosis, pelvic inflammatory disease (PID), genetic disorder like Turner syndrome. Common causes include unhealthy lifestyle habits, genetics, infection, hormonal imbalance, structural abnormalities. Next question is, explain in vitro fertilization with diagram. In vitro fertilization (IF) is a type of assisted reproductive technology (ART) where egg is fertilized outside the body in a laboratory dish. Here is a step-by-step explanation with diagram. IVF process. Ovulation induction. Medication stimulates ovaries to produce multiple eggs. Egg retrieval. Eggs are retrieved from ovaries through a minor surgical procedure. Fertilization. They are mixed in a laboratory dish allowing fertilization to occur. Embryo culture. Fertilized egg embryos are cultured in a specialized medium for 3 to 5 days, and embryo transfer. One or more embryos transferred into the uterus. Diagram. First में ovulation induction होती है, then agri-tribal surgical procedure, then sterilization, and then embryo culture 3 to 5 days, and then implant embryo transfer into uterus. So students, हमारी लाखों से भी कंप्लीट हो चुके हैं. होपफुली आपको समझ आगे होंगे. If you have any doubt, you can ask in comment section. Please like, share and subscribe my YouTube channel. Thank you.